In a light microscope, visible light is passed through the specimen and then through glass lenses. This is an example of a specimen under a light microscope. A scanning electron microscope is a type of electron microscope. An electron microscope focuses a team of electrons through the specimen or onto its surface. An SEM is useful for detailed study of a topography of a specimen. A transmission electron microscope is used to study the internal structure of cells. This demonstrates the ranges which you can see with an unaided eye, the light microscope, and an electron microscope.
The surface to volume ratio is represented with these boxes. As a cell increases in size, its volume grows proportionally more than its surface area. These boxes are proportionate in size, but the larger box's volume is greater than its surface area. The mitochondrion is bound by a double membrane. The inner membrane has foldings called cristae. It is important for cellular respiration, and it produces ATP after organic material is consumed.
Chloroplast has two membranes around fluid stroma. It includes thylakoid sac and integrana. It receives sunlight, then goes through photosynthesis and produces ATP.
Peroxisomes are specialized metabolic compartments bounded by a simple membrane. They contain enzymes that transfer hydrogen ions from substrates to oxygen, producing hydrogen peroxide as a byproduct.
H2O2 is then converted to H2O by another enzyme.
The cytoskeleton is a network of fibers extending throughout the cytoplasm. It gives support to the cell and maintains its shape. It contains microtubules, microfilaments, and intermediate filaments. Microfilaments are the thinnest filaments that give the cell its shape and enable cell movement. Microtubules are the thickest fibers made up of the globular proteins. They participate in the intracellular transfer of organelles and vesicles. Intermediate filaments are made of a variety of proteins, including keratin. It provides mechanical support for the plasma membrane and does not participate in cell motility.
Selective permeability is the process by which a cell exchanges molecules and ions with its surroundings. Cell membranes are selectively permeable and substances do not cross the barrier indiscriminately. A cell is able to take up small molecules and ions and exclude others. Transport proteins. Polar molecules and ions generally require specific transport proteins to cross the membrane. ATP was needed to cross the gate.
Osmosis is the diffusion of free water across a selectively permeable membrane, whether artificial or cellular. In a hypertonic solution, the outside has a higher solute concentration. The cell shrinks, which is shown by the balloon shrinking. In an isotonic solution, if the concentrations are equal inside and outside the cell, the balloon is the balloon or the cell is at equilibrium. In a hypertonic solution, the outside has a lower solute concentration. Water enters the cell and the cell bursts, which is shown by the balloon bursting.
Passive transport, also known as facilitated diffusion, is where a transfer protein speeds the movement of water or a solute across a membrane down its concentration gradient.
In active transport, specific membrane proteins use energy in the form of ATP to move solutes against their gradient. It enables a cell to maintain internal concentrations of small solutes that differ from concentrations in its environment.
This transport system pumps sodium and potassium ions against steep concentration gradients. It is an example of active transport. In order to pass the ions through, the pump needs ATP. For every 3 sodium ions that go out of the cell, there are 2 potassium ions that go into the cell. The pump continues to need ATP to change its shape to that of the sodium and potassium ions.
In co-transport, a transport protein, a co-transporter, can couple the downhill diffusion of a solute to the uphill transport of a second substance against its own concentration gradient. Bulk transport is when large molecules such as proteins and polysaccharides, as well as larger particles, generally cross the membrane in bulk, packaged in vesicles. Bulk transport across the plasma membrane occurs by exocytosis and endocytosis.
Form of bulk transport in which materials are transported from the inside to the outside of the cell membrane.
Endocytosis is a general term for the various types of active transport that move particles into a cell by enclosing them in vesicles made out of plasma membrane.
Phagocytosis, also called cell eating, is a form of endocytosis in which large particles are transported into a cell. Pinocytosis, also called cell drinking, is a form of endocytosis in which a cell takes in small amounts of extracellular fluid.
Receptor-mediated endocytosis is a form of endocytosis in which receptor proteins on the cell surface are used to capture a specific target molecule.